Why? Because it indicates very clearly that complex 1 has 2 enzyme active sites. So the NADH oxidoreductase part, oxidoreductase means you're transferring electron. This enzyme activity is occurring right here. That's a catalytic site. That site only recognizes NADH. And it's going to pass electrons down this circuit. And people always ask me, why does it pass 1 electron instead of 2? Or why does it do 1 at a time? My why questions are difficult to answer. Our best guess is that nature has found this to be more efficient in terms of getting energy out. The CoQ oxidoreductase part is happening over here. They don't have the exact site. But this is where the electrons here, this line has no bearing. The electrons are being enzymatically catalyzed onto Q. So there is a very specific site over here somewhere. So there's 2 enzyme active sites. Is that clear? Complex 2 is pretty big. It has 4 subunits. And this end out here is where succinate dehydrogenase is located, which is the Krebs cycle enzyme that we talked about. But there's 2 enzyme activities. One is succinate dehydrogenase here, and the other is, again, an enzyme active site here somewhere. Again, it's a 3-dimensional object pressed onto a 2-dimensional plane, where Q is having electrons put directly onto it by this enzyme. So the enzyme makes things very fast, and it makes it very specific. Yes? Another why question. Why questions are good. Okay. The cell is trying to squeeze out as much energy as possible. So in doing the step where it goes from succinate to fumarate, it's getting some electrons off, 2 electrons. But notice that when these electrons come in their blue line here, and this is all of these blue lines that are in the protein come with giant question marks. Because within these complexes, there are certain points where the humans can see an electron just went by. Because those points change their spectrum. So we can go, oh, something just went by. But during most of this circuit here, these electrons, there's 2 complexities. One, the electrons are going through the backbone of the peptide protein and through the R group. So that's one problem. The second problem is, at this level, we're going to nitpick it apart. You honestly have to talk about the electrons moving as wave functions. They're not part of it. Okay? So you got to get rid of that one. So they're moving as waves through the circuit. Cool? And you can never say that the wave goes exactly this way. All right. Getting back to here. The cell must make fumarate on its way back. Where was that question? Yeah. It has to make fumarate to get back to oxaloacetate to run the Krebs cycle. So in that process, it must make OAA, and if it can take 2 electrons and send them in this way, notice you only get 6 protons. But that's still better than 0 protons. So I would look at it that way. It is, okay, what we can do is say it's always the same enzyme. So down here, succinate dehydrogenase is here. All the other enzymes of the Krebs cycle are floating around in here. Okay? Now, 50 years from now, they may find that, oh wait, fumarase is actually loosely associated right here. I wouldn't be shocked, but right now we don't know that. Where you find a different version of the enzyme is if the enzyme is necessary out here in the cytosol. So the same enzyme out here will be a different isozyme than this one here. You follow me? Because the environment out in the cytosol is different than inside the matrix. So here, I want you to see from this hi, how are you slide, that when succinate passes electrons through, the electrons are missing the production of 4 protons over here. The electrons coming off of FAD are not as energetic, hence they can't be used to pump 4 extra protons across, and that's why the FAD from succinate only produces 1.5 ATP, whereas this guy down here can produce 2.5 ATP. Well again, in a laboratory, when you break open a cell and you isolate an enzyme and you find, wait, there's 2 peaks, there's 2 different enzymes. You go, oh, this is nice, I like this. And now I'm going to purify each one, sequence it, and find out they truly have different amino acid sequences, and yet they both catalyze the same reaction. So then we can make antibodies to them, label the antibodies with radioactivity or fluorescence, most people use fluorescence, and then take cells and go, this antibody lights up in the cytosol. This other guy, his antibody lights up inside the mitochondria. Right on. Now we start to understand why life has different isotops. Okay, let's finish. This guy here, the guy with 2 caverns, complex 3, is going to transfer electrons onto cytochrome C. So there's an enzyme active site right up here. There's really 2 active sites. One is right here where the transfer of electrons, one at a time, onto a cytochrome C. And then where Q comes into the cavern, there's 2 caverns, so there's room for 2 different co-Qs. Where it binds is at an active site that catalyzes the transfer of one electron from Q onto this pathway. What they're not showing is the other blue line that comes down here. I guess they were afraid of scaring people, but to show them the second blue line. But you'll see that on Monday. Okay? So we get scared on Monday. How about that? So, what I can say for right now is that complex 3, it's got these 2 caverns, and it's got a weird protein. My hand here is the protein, and the head of the protein, the domain, kind of looks like a camera. And so one cavern has got this weird protein. The other guy has got its brother, and they go up and down like this, like a 2-cycle engine. Because there's only one binding site for cytochrome C. There's only one landing spot, and yet there's 2 caverns. And so they apparently work in opposite flow. Cool? Well, it'll be clear. This is why we do research. You go, you can't believe there's 2 caverns here in complex 3. You got this weird protein going up and down. What's with that? Well, it's only been doing that for like 1 billion years, so we're catching on now. Cytochrome C, 13,000 molecular weight, too big to get through those pores, not shown in this diagram, that only allow things that are about 5,000 molecular weight to get through. And then over here, complex 4 is where the chemistry is occurring, where oxygen is being reduced to water. We'll pick that apart, and that will probably be the hardest lecture of the entire course. We'll look forward to that maybe on Wednesday the next week. A lot of subunits, again, most of it you don't know what they're doing. But one of the interesting things is that when the protons go across this complex here, this is a huge simplification. It turns out in complex 4 there's 2 channels, and the protons hop through the channel. There's no hopping anywhere else in the diagram. So even though protons are moving in complex 3 and complex 1, we know for sure that things are different here in complex 4. So when I say hopping, I mean if the membrane was right here and I'm a proton and I come in, the guy who comes out on the other side isn't me. Protons are hopping incredibly fast, much faster than diffusion. So light has come up with some interesting ways of getting protons across. Those 10 protons are not getting across the same way in all the different complexes. Okay? So this is cytochrome C oxidase. It's taking one electron from cytochrome C. There's an enzyme active site, and now the electron goes through the circuit and gets added to an oxygen molecule. And so really, oxygen doesn't exist as a half O2. It's going to be 2 oxygens, OO. So to reduce it to water, it's going to take 4 cytochrome Cs, docking one at a time, because they can only bring 1 electron. This whole process here, going from start to finish, takes about 1 millisecond in the cell. So this is incredibly fast. What makes it fast are those enzyme active sites that catalyze things. The current flowing is instantaneous, but the enzyme active sites help make the whole process very fast. Okay? So when we study this, you can say to yourself, man, it's amazing. This whole process is in 1 millisecond. How can they possibly study this? It took a long time. Okay, in this process, we've got a number of iron-sulfur centers. You've seen this one before. No, Dr. Hill, I haven't. Well, I'm worried about the midterm math. This is the same iron-sulfur complex, a 4-iron, 4-sulfur complex, in which enzyme in the Krebs cycle? Aconitase. But there are other versions. This would be a 2-iron, 2-sulfur. They don't count the sulfurs here with the cysteines in the nomenclature. Just like there are cysteines here with sulfurs. They're only talking about what's in the cube, or here what's in this rectangle. And over here is a 1-iron complex. So these guys have different redox potentials, depending on how many irons and sulfurs are involved, and also dependent on the protein itself that's binding to it. They act as one. So these guys here are acting as one. The other type of iron-sulfur complex that we have to talk about is a risky iron-sulfur, named after the person who discovered it. The only difference is it's a 2-iron, 2-sulfur complex where the cysteines here, 2 of them, have been replaced with histidines. And so we'll just block this part out and write histidine. This histidine can coordinate through its side chain. The side chain, the imidazole group, one of the nitrogens has 2 electrons on it. It can form a bond, just like the nitrogens in a porphyrin ring. So these are the players in terms of part of the complexes that humans can actually see the electron was just at. So in the electron transport chain, these are all involved in electron transfers. They're doing 1 electron at a time. Okay with that? Can we change the slide? Kind of. So life uses these things, irons that are not in heme. So that brings us to this. if I showed you this, you could say, yeah, those look like these, okay? We're not memorizing. But the point is, over here, first of all the pink, Leininger is showing you the conjugated double bond system. This is why it absorbs light. This heme here is the one that you're familiar with if you had BIS 102 where we discussed myoglobin and hemoglobin. This is the heme that we're familiar with. It can bind oxygen at the central iron. These are other types of hemes. And notice that this one here has a covalent linkage here to cysteines. This one down here has a long hydrophobic tail. The point is there's different versions of hemes, and their ability to either pass an electron or to bind oxygen depends on the protein that they're part of too. So these, again, today's just, hi, how are you? These are some of the players. Okay? I'll be honest with you. Okay? Nothing happens though without the protein. So these hemes and iron sulfurs are nice, but each one of these things is held perfectly in position by the subunit that it's embedded in. Okay? So don't get overly enamored with these guys. They're just pieces in protein. It's the proteins that are necessary. And our last slide here is showing you what ubiquinone, also called Q. Ubiquinone just means, it comes from the word ubiquitous. It's everywhere in the cell. And you may call this molecule CoQ. The point is, first of all, you're never going to draw the structure. If I drew the structure, you could say, oh, that's CoQ or Q or ubiquinone. It has two properties. One, it has this moiety here that very safely can carry one electron. And so here, that electron is shown as a dot. But due to resonance, you could spread the dot over many different positions on this molecule. Are you okay on that? So it's a safe way to house a free radical. And the other important property is this portion here, these 5 carbons, this is an isoprene unit. Maybe you saw this in organic chemistry, I don't know. But it's a common molecule, and there's 10 of these. This is a very long set of isoprene units that make for a very hydrophobic tail. So this molecule here is never getting out of the inner membrane. It's locked inside. So on comes an electron and a proton, and then it can take another electron and proton safely. So the fully reduced form, where R here is this long hydrophobic tail, this is a carrier of 1 or 2 electrons and 1 or 2 protons. This guy is going to be involved, as we saw in a previous slide, in donating of protons across the membrane as part of the proton gradient. So we know for sure that this guy's involved in complex 1 and complex 3. He's not a player in complex 4. That complex 4 is where the proton popping is occurring. Cool? It's Friday. Let's call it a day. Have a nice weekend.